Hi, hello, welcome to the channel Meekosam Health and Education. Today I am discussing about the cell wall. The cell wall is a thick wall which is present in both plants and bacteria, but not in animal cells. Yes, what is the structure of or outer structure of cell wall? Let us discuss. Yes, the cell wall is made up of mainly three layers: primary cell wall, secondary cell wall, and middle lamella, which is primarily composed of the three important segments: cellulose, lignin, and pectins. Yes, these are the class of carbohydrate molecules which are comes under polysaccharides. Cellulose, homopolysaccharides; lignin and pectins, heteropolysaccharides. Yes, right. The primary cell wall is primarily made up of the cellulose fibers, which is the what we call primary, means it is the first wall characterized by cellulose carbohydrate fibrils. Yes, the next one, the secondary cell wall, it contains the pectins. Yes, pectin fibrils are present in the secondary cell walls. Most probably in plants, the secondary cell wall is characterized by lignins. Okay, secondary cell wall is characterized by lignins. Yes, lignin is a rigid carbohydrate. That's why it gives the mechanical strength to the plant cells. Yes, lignin is characterized and gives the rigidity to the plant cell. Yes, coming to the last one, middle lamella. It only comprises pectins. Okay, it only comprises pectins. It connects with the outer periphery of other cells. That means it can react with other adjacent cells. It is the role of middle lamella. This is about plants cell wall material. Cell wall in bacteria. Yes, bacterial cell wall is quite different from the plant cell wall in the composition. What we call, it comprises mainly the portion of cell wall is peptidoglycans. Yes, the peptidoglycans are the peptidoglycan comprise, that means peptide part and glucose part. That means glycan part. Okay. Yes, this is peptidoglycan part, which comprises mainly two types of chemicals, what we call N-acetyl muramic acid and N-acetyl glucose amine. Yes, the N-acetyl muramic acid and glucose amine, NAM and NAC, which forms the peptidoglycan layer, which comprises cell wall. Just like this, the repetitive units of NAM and NAC, which comprises peptidoglycan. Yes, in some bacteria it is very thick, and in some bacteria it is thin. Yes, thick comprises by means of most probably 70% of a peptidoglycan. Thin, whereas based on the content of peptidoglycan, the rigidity or the strength or whatever it may be, the pathogenicity of that particular bacteria or prokaryotic organism is defined. For example, if the bacteria or prokaryote cell is having more than 70% of peptidoglycan, it is very hard to kill the outer agents or the chemical agents or detoxificants. Whereas the thinner bacteria, that means the 30% only comprises 30% of peptidoglycan, is very easily exposed to the disinfectants or whatever. That means it easily killed. Based on the type of this cell wall peptidoglycan, the bacteria was classified. That means into two major groups, what we call gram positive and gram negative nature. Okay, this topic we will discuss later. Okay, yes, this is the type of cell wall present in bacteria. Yes. Coming to the next cell organelle, what we call centrioles. Yes, the centrioles are which is comprising the chromosomal parts, centrosomal parts, only found mostly in animal cells abundantly. During cell division, the centrioles helps to formation of spindle apparatus. During cell division. Either it may be the mitosis or meiosis. Cell division. what we call division of cells is known as cell division. That means one cell divided in when it forms the next generation. This process is known as simply cell division. Yes, there are two types of cell divisions majorly. We found mitosis and meiosis. Yes, instead of that, the third category is there, amitosis. That means other than mitosis and meiosis is known as amitosis. This is takes place only in the primitive organisms like primitive bacteria and unicellular prokaryotes. Yes, unicellular prokaryotes. For example, these are the cellular division processes which is takes place in primitive organisms. Comes under amitosis. Yes, coming to the mitosis process. Yes, what we call somatic cells. In somatic cells, the division is takes place by mitosis. Why we call this mitosis? Means the one cell divided into two. That means 2n stage. The diploid nature. The diploid nature was conserved. This type of cell division is known as mitosis. Especially it is takes place only in somatic or body cells. Okay. Then coming to the next category, meiosis. Only it takes place in gametic cells. Reduction, division. That means chromosome number reduced to half. That means 2n. 2n is reduced to half. That's why it is a reduction in chromosome number. We call reduction, division or meiosis. Yes. Coming to the operators of cell division. Yes. What are the requirement? Basic requirements means cells, and then the next one nucleus, which comprises chromosomes. Spindle apparatus. I am already told. The centrioles forms the spindle apparatus. The next one is cell plate apparatus. That means it is a plate-like structure. When a particular cell divides into two, it is a plate-like structure. Forms or construct the cell. Then later it was divided into two equal cell division. What we call one cell divided into two cells by means of two types, mitosis and meiosis. Amitosis already we will discuss. We will discuss later amitosis. Yes, the karyokinesis nuclear division, cytokinesis cell division. These are the two steps involved in cell division. Yes, in mitosis there is four different phases: prophase, metaphase, anaphase, and telophase. Okay, these are the four distinct phases present in mitosis. Whereas coming to the meiosis, it is in the two-step process. What we call the first one is meiosis 1 or reduction division. The second one is meiosis 2 or mitotic-like division. These are the phases present in meiosis 1 and meiosis 2. First of all, we will go for mitosis. Prophase. This is the first phase of cell division, which the chromosomal parts, that means chromosomal apparatus, is started for division after the DNA replication. DNA is the major component of chromosomes which passes heredity. Okay, after the completion of this DNA replication, the cell was divided. First phase is prophase. Okay, after completion of this DNA replication, prophase starts. That means chromosomes are starting for division. This phase is called prophase. The next one, metaphase. Here we found the formation of cell plate. Okay, cell plate forms. That means constriction at the center part of cell wall or cell membrane. Okay, it is called metaphase. The next one, anaphase. Chromosomes are pulled towards the opposite poles by the spindle apparatus. This phase is anaphase. This process continues till the end. It is called telophase. Okay, these are quite similar, anaphase and telophase. In anaphase, the chromosomes are separated towards the opposite poles. This process continues up to the cell, which is divided into two equal parts. This process, this stage is known as a telophase. After completion of this division, we got to distinct cells. That means chromosome number, for example 2n, yes, this is also 2n. That means conjured. Okay, the chromosome number is conjured 2n. If it is n, we also found here n, but not possible. Why? Because the all somatic cells are in 2n stage. Okay, chromosome number is conjured. That means it is called mitotic. The next division, meiosis. It is takes place in two phases, meiosis 1 and meiosis 2. Reduction division. That means chromosome number 2n chromosome number is reduced here. n. Okay, chromosome number is reduced. In this there are 4 different phases: prophase 1, metaphase 1, anaphase 1, and telophase 1. Condensed. This is the characteristic feature of prophase 1 leptotin stage. The next, zygote stage. Yes, this is a stage what we call flower bucket stage, or bouquet or bucket. The chromosomes are arranged like this, flower bouquet or bucket. This is a zygote phase. Coming to the next phase, packaging. A very well evolutionary process takes place, what we call crossing over. That means chromosomal parts are equally exchanged. Okay, equally exchanged, crossing. Due to this crossing over, evolution is carried on. This takes place in the packing stage of prophase 1 of meiosis 1. The next, diplotin. This process continuation. Crossing over is continuation to the next phase. After that, the cell was starting to divide. That means in diakinesis, all this process takes place in nucleus. After that, this diplotin stage, the diakinesis is characterized by disappear of nuclear membrane and slowly the nucleolus also. Disappear of nuclear membrane and nucleolus is characterized by the phase diakinesis of prophase 1 of meiosis 1. After that, the cell enters into the metaphase 1 stage. Yes, what we discussed already. Metaphase 1, cell plate formation is there. Anaphase 1, spindle fibers or spindle apparatus pulled the chromosomes towards opposite poles. The next, coming to that process continuation, it is called telophase. After that, this telophase 1 of meiosis 1, we got two cells. Which are n stage. That means chromosome number reduced. That's why it is meiotic or reduction division. So two cells are formed, which are in haploid stage. After that, this completion of telophase 1, the cell was entered into meiosis 2. What is meiosis? I am already right here. Mitotic like division. What is mitosis? Chromosome number is conjured. Here chromosome number is n. Here also n. That means two daughter cells. These again divides and forms into n. This also divides and forms into n. That's why meiosis 2 is also known as mitotic like division. It also has 4 phases: prophase 2, metaphase 2, anaphase 2, telophase. Quite similar to these processes. Okay, After completion of the total meiotic process or meiotic cell division by both meiosis 1 and meiosis 2, we got 4 daughter or haploid cells, which all are in n stage. That means haploid stage. So we started with 2n. These are divided into nn. That means diploid, haploid. These are gametic cells. So gametic cells chromosome number is reduced. Okay, chromosome number is reduced.